All right. So yesterday we were, refreshing our memory about how a eukaryotic gene looks, right? Different parts. Now, okay, the slide is here. If I had a way of blocking it, I can immediately ask a quiz and see whether you remember. So where will you find the cap? 5 prime? Anywhere in the UTR? At the very beginning of the UTR. Okay. So what will be the relationship of transcription start site with relation to exons, introns? No, it can be either. Okay, so the 5 prime UTR can very well be starting at the transcription start site, right? So it can be in the exon too. Alright, so we will do more quizzes later on this. So right now we will continue on this. So today what we are going to do is mostly focusing on molecular biology techniques, some of which you may have learnt in other courses and some of them may be new, because some of them are at whole organism based techniques and that you may not have heard elsewhere. So essentially what the main focus is what are the techniques by which we try to understand differential gene expression. Okay. So that is going to be the focus for this lecture as well as the next one. So this is continuing on refreshing our memory of the eukaryotic gene structure. So the previous one was a cartoon, this is an actual sequence, okay, of the beta globin gene. So you have the upstream promoter elements like the starter box here, and this the minus 70 sequence. So these are there in most of the promoters, that is why they are part of the core structure here. So this is the sequence that signals the cap addition. And you see this is right away is an exon. The colored ones are exons, the gray are the introns. So, okay. So you see that the UTR is an exon and there you have the cap. Start codon ATG, then this is the actual nucleotide and the translated amino acid sequence, then you have the beginning of an intron and end of an intron and then so on. Then at the end you have the stop codon, then you have the UTR, 3 prime UTR, right. So then you have the AAT-AAA, this is a canonical polyase signal, but there are other variants of this that also work as polyadenylation signal. So they help in identifying the cleavage site also. So actually the machinery that does the cleavage, what the cleavage here means is the pre-mRNA nascent transcript can be longer at the 3 prime than the mature one in the cytoplasm having the poly A tail. So at the 3 prime there is a cleavage that happens. So some amount of the transcribed mRNA is removed and to the remainder you add the poly A tail. So that itself is a major complex and highly regulated, it is as big as a ribosome. Okay. Another such major complex is the spliceosome, okay, the splicing machinery. So these are major protein complexes that operate. So next we are looking at the transcription itself. This is the first step here. So you have the transcription. So you have, as soon as the nascent mRNA comes out or nuclear RNA, so you have the cap added, trimethyl G cap. At that stage splicing is not done yet, okay, that is what I want to point out. These additions help in protecting RNA, okay. So but I am not sure whether that is the reason why that step is first, but these additions at both ends happen before splicing. So then you have processing, which is essentially splicing, then you have the messenger RNA or mature mRNA. So this comes into the cytoplasm where it gets translated, then you have the protein chain and then that has to undergo two things, one proper folding to the native conformation and also post translational modification. So here for example this prosthetic group gets added, subunits of, this is a 4 subunit protein, it is a heterodimer. So beta globin, alpha globin, 2 each come together to make the functional molecule. So all these steps put together is what you call as gene expression. Okay. So, do not assume only transcription is gene expression and this is something like protein level. The whole thing is gene expression. When you talk gene, the definition of gene is biological function, that is the phenotype. So, protein is obviously responsible for it. So, we do not distinguish these steps. All the steps from the chromatin structure, modifications to the chromatin structure, like from heterochromatin to euchromatin transformation that happens due to removal of methyl groups and addition of acetyl groups and specific methyl groups to H3 tail. Starting from there to the post translationally modified active or inactive protein. So, up to that the whole steps are called gene expression. Each one is one step in gene expression. So, now we know what is a gene and what is its final functional form. So, now let us look at, again we are refreshing some of the steps, we are still not getting into differential gene expression. For that we want to familiarize with what happens, so that we will know where regulations can happen. So, the first thing is we are going to look at the transcription initiation itself. So, transcription initiation, so this cartoon goes with some details, but the summary is that a set of proteins have to bind in a certain sequence to enable the RNA polymerase to start transcription. So, the initial assembly and recruiting the RNA polymerase to the promoter is what you call as transcription initiation. So, here you have TF2D, transcription factor 2D. So, that has to first bind, it binds to the Tata box, then recruits 2A, 2D, 2A, and then you have B and H. So, these clamp like structures they add, and then only you have Pol 2 recruited, and to the Pol 2 you have the E and F already binding to it, and when it binds, this carboxy terminal domain CTD plays a major role in regulation. So, that is bound to the 2D structure, this main molecule, and so that is how it is. So, this is already set, but it is not go. Okay. So, that requires phosphorylation of CTD. So, to phosphorylate or not to phosphorylate is a step there. If you do not phosphorylate, no transcription, although everything has bound and it is all ready. So, that is a step where regulation often happens. So, once this gets phosphorylated, there are certain serine residues that are critical for the phosphorylation. So, oftentimes developmental biologists use those phosphorylated serine specific antibodies, phosphorylated CTD specific antibodies, to measure global level of whether transcription is happening in a given nucleus or not. Okay. So, with those phospho-specific antibodies, CTD specific, for phosphorylated CTD specific antibodies, if you do not detect a signal, probably there is no mRNA transcription happening. So, remember we are talking only mRNA, but it is because it is Pol 2 specific antibody we are talking about. So, other transcription could be happening. So, once it is phosphorylated, then it is released from TF2D and it is ready to elongate. So, initiation then elongation. Okay. So, this is initiation step and this is elongation, and even in elongation it can be paused sometimes. Okay. So, those details we are not going to get into, but this is good enough to know that this is a potential step for regulation. Alright. So, now so we are done with those basics. So, we are going to now look at a series of experiments that help us to identify sequences in the promoter region and as well as elsewhere in the vicinity of gene that could be contributing to transcription. One, and second, we are also going to look at how does one find transcription factors, that is transacting factors like proteins. Okay. The DNA sequence itself we are going to say cis because it is in the same molecule as the open reading frame, and protein is encoded elsewhere and it comes as a different molecule, different from that piece of DNA where you have the gene. So, therefore, it is called trans. So, when you say transacting factors, you are talking about factors other than a given gene sequence that influences that gene expression. Okay. So this is a very commonly used assay. People use it for multiple contexts where you want to test nucleic acid protein interaction. Okay. So right now we are looking at DNA protein interaction to find out the promoter elements that may be involved in interacting with a specific transcription factor. So these transcription factors essentially, like you already saw a base. So these molecules, this shown in this blue color, so these are aiding in recruiting the RNA Pol2 to the promoter, and they are transcription factors. So these factors listed here and named, these are there for every gene. These are core transcription factors, and there are gene specific transcription factors that we will learn later. And so how do we test whether a given such transacting factor interacts with a given element of DNA or not? So to test that is what we use this experiment called a gel shift assay, or gel mobility shift assay, or gel retardation assay. So there are multiple names for it. Most commonly used term is electrophoretic mobility shift assay, or EMSA, or EMSA. Okay. So some labs call EMSA, some labs call EMSA, some labs do not even say that, they say mobility shift, gel shift. Okay. So it is extremely simple technique. So what you are actually doing is you take a radio labeled version shown here in this whatever color that comes on that screen. Here for me it looks purple. So you take radio labeled version of the DNA fragment you want to test whether it interacts with a given protein. Then you incubate with protein. It may be either purified protein or it is a cell lysate where the protein may be present. So you incubate with it and then you run on a gel. If the protein binds to that DNA fragment, then the complex size is bigger than the individual nucleic acid, so the mobility is reduced. Since in the gel you see it as a shift, an upward shift, you call it as gel mobility shift assay, or since it retards the mobility of the nucleic acid, you call it as gel retardation assay. So all those names are really valid names. So that is how you see it. So here in this particular case, PAX6 is a transcription factor about which we will see some more, and without that the free nucleic acid, we call it as free probe. So free probe for a given time in a given conditions of gel, for example gel pore size, etc., it moves a certain distance, and when the transcription factor is added which binds this particular DNA fragment, its mobility is shifted. Since it is radio labeled, you can detect it by autoradiography. Okay. So this is a very versatile and very useful experiment. In our lab we primarily used it for finding RNA binding proteins that interact with 3' UTR and regulate translation. So that is the context in which we have used. So therefore it is useful for nucleic acid protein interaction in different contexts. So in 3 you have PAX6 added, so it shifts, alright, and in lane 4 also PAX6 is added. It is a control experiment. So here what you are doing is, when it binds, then there are two alternative explanations possible. Maybe it is a protein that would bind any nucleic acid, okay, non-specifically, it may not be sequence specific. To test that, what you do is you add a large molar excess of the same nucleic acid fragment but without radio labeling. Okay. So now what will happen, depending on what is the molar excess, let us say I have 10-fold molar excess or 100-fold molar excess, then this intensity will go down. Here there is only one because it is a cartoon and just showing you the the scheme, but in an actual experiment we will have 2-fold, 4-fold, 10-fold, 100-fold, like that serially we will be increasing the unlabeled same nucleic acid. So that would also have equal tendency to bind. It is more like a competitive inhibitor. So now you have the band there, but that is not having radioactivity, so you do not detect it. Okay. So that, and then another control which is not shown here is you add similar molar excess of similar number of nucleotides, okay, similar length of the nucleic acid, but of a non-specific sequence, and that will not compete out. If it is sequence specific binding, same sequence unlabeled will compete out the radio label there, but a different sequence will not compete. So, even in its excess, this protein will still bind to the that particular sequence. Alright, so the next one is DNase protection assay. So, this you do it, see this is looking sort of not familiar from our facial expression, I can detect that. That is because you are no longer doing DNA sequencing by this method, but otherwise like when I was a student, this is a routine gel. So, this kind of image you see every day if you are doing DNA sequencing. So, on those days we did not really do whole lot in developmental biology. So, these molecular biology experiments is what most other people did. So, this is how you do DNA sequencing. So, you have radio labeled oligo to start with, and in each reaction you have dideoxy of that nucleotide. So, you will have 4 lanes. So, you will run 4 lanes on a gel, and then you look at in which lane you have the smallest fragment, and from there progressively you count upward, and that is how you get the DNA sequencing. So, this is one such DNA sequencing gel, but here you are not sequencing the DNA itself. So, instead what you are doing is the DNA is incubated with a protein that binds to some region. So, this is really large fragment compared to what is used in gel shift assay. Okay. So, here you are actually trying to find which sequence in a larger sequence binds to the protein. Then once the protein binds to the DNA, you add a nuclease, a controlled nuclease treatment you do. So, now what happens is the fragment that is protected by the first protein that bound to it protects that sequence, and the rest of it gets cleaved. And when you run a gel, since it is a controlled digestion and you have partial digestion for every nucleotide length. So, in the control lane you see bands for the entire length, because at every place there could have been digestion. But if you look at this portion in these 2 lanes, and then here you see bands missing when you have added PAC6. Okay, so, if you compare this with this as well as this region in the control with this, so indicating that PAC6 binds to that region. So, essentially you are testing whether your protein protects, which portion of a DNA fragment does it protect from a nuclease cleavage. So, this is also done with RNase protection also to find part of the RNA where an RNA binding protein binds. So, this is DNase protection assay. This is also called footprint assay, okay, because it is like a footprint, the protein's footprint. So, now before we go into next set of techniques, we are going to look at a part of the gene, okay. So, regulatory element, it is not really the coding part, and it can be anywhere actually, it need not be always in the promoter region, it can be at considerable distance, it can be downstream of the transcription initiation side as well. So, that is what we are going to look at, and they are called enhancers. So, what are enhancers? The first point tells you they control efficiency and rate of transcription. Okay. So, they may increase the rate, or in some places they very strictly specify spatial or temporal expression of a gene. Okay. So, that is shown here in this is the PAC6 promoter cartoon. So, these orange colored bars or bands are the exons, okay. This red one is the promoter itself, then this green is the enhancer, and you see enhancer present here in 1, 2, 3, 4 fragments, 4 parts, and one of them is actually in an intron, okay. This is an exon and this one exon, and this whole thing is intron. So, it is part of an intron too, okay. So, these enhancers, as I told you, they control efficiency and rate, and sometimes very strict temporal and spatial regulation. and here we see such a spatial regulation information. For example, this enhancer between exon 4 and 5, this confers expression of this gene, that is PAX6, in retina. Okay. And this particular exon here, just before the transcription start, is a neural tube enhancer that makes sure this protein is expressed in neural tubes, and the other one lens and cornea. If you do not have it, it is not going to express there, and the other one, that upstream most, pancreas specific. So each one of these 4 have 4 different tissue specific expression of this gene. So you might have the core promoter, core transcription factor, everything, but you are not going to have transcription if you do not have these enhancers and then those enhancer specific transcription factors. Okay, so we will see them one by one. So this one, this actual sequence tells you in a part of this pancreatic enhancer sequence where you see binding sites for 2 different transcription factors. Okay, two different transcription factors bind to that. So a little bit more detail on that we will see 2 slides later. Okay, there are many ways by which one identifies enhancers, and here just one easy to understand and very powerful, historically had been very useful technique called enhancer trap. So what you are actually doing is you are having a core promoter driving a reporter gene, all of it embedded in a transposable element. Okay, so this is useful in an organism where natural endogenous transposable elements exist and they hop on and hop out depending on the context, whether the transposable element is active or not, depending on that they will move around the genome. And usually transposable elements to get inserted, they need repetitive sequences, and wherever that is there they will go. So now if you use the transposable elements as a vehicle to carry this reporter with a core promoter, meaning no transcription factor binding accessory sequences like no enhancers or anything, and now you provide the context in which transposable elements are active, and then you stop it and then look at all the different insertions, kind of mutagenesis using transposable elements. Okay, instead of some other mutagen, here you are using transposable elements. So in different individual there of that organism, you will have transposable element inserted in one part of the genome or another part of the genome. So now suppose this transcription, this transcriptional reporter gets inserted in the vicinity of an enhancer, as you see in this picture here. Like here it has come next to an enhancer, okay, and this enhancer normally controls the expression of a gene downstream. So this is the gene here, and now this enhancer influences the expression of this, then this gets expressed where this gene is normally expressed, and then you can detect it this way. Okay, this is a Drosophila embryo, so where you see the expression pattern. So essentially you are trapping an enhancer with the help of a reporter gene. So the reporter gene has a weak promoter, it just has the core basic one. So it is not going to express on its own without coming under the influence of an enhancer. So when you do random insertion, if it gets inserted near an enhancer, then it expresses where that enhancer normally activates transcription, and that is how you find, oh, this enhancer works in this tissue. So later that can be verified by other methods as well. Reporter gene is a laxert here. So this is done in an era long before GAP was discovered. Okay, so that you will find out by sequencing the flanking region. So that will not be difficult, but the point is you have trapped an enhancer. Okay, so the primary goal, again these are all coming from the thought process of genetics. Okay, in genetics you do not worry about the molecule first, you first find out, did I hit a molecule that is responsible for a function, then you can search and find the molecule. That is lot easier than having a bucket full of molecules. Like you can take any organism and crush it and send it to a company and they will give you all the proteins produced there, all the mRNA produced there, but what will you do with it without connecting to the function? Okay. So, normally we call that you get a big laundry list, but you do not know what to do with it. Okay. There are many such lists that are hanging around for the last 20 years without people having made progress. This is not to belittle the usefulness of that, this is only to highlight the importance of directly connecting to the phenotype. Because it is very easy to do an inverse PCR and sequence the neighboring sequence, because you know this sequence. Okay. So, you can digest with restriction enzymes, and some might cut somewhere here and there, and under a very dilute condition, if you ligate it, intramolecular ligation will happen. So, it will get circularized. Then you take the sequence you know, then you use primers going in the opposite direction, then they will sequence the flanking PCR amplify the flanking region, then you sequence, then you will know where it is. Okay. So, that is how you identify. But the primary goal here is to find, will I get a tissue specific activation? So, that means I am in the vicinity of a potential enhancer. So, that is the goal of this. So, these were done in the mid 80s to late 80s period. So, the next one is the same thing, but helping you to determine, like for example, if you know an enhancer and then you want to find out what are all the different places where that enhancer works. So, you can make reporters under the control of enhancer and look at it. So, this is an older assay method where it is lag Z, and then you see its expression for primarily in the central nervous system here and muscle specific expression. And here what you are seeing is lens crystalline enhancer, and you see where it is expressed. So, this is the image I particularly like because it shows you tissue specific control so clearly, right. So, there are a lot of such things. Yesterday in the lab we had some images where the gene is GFP is expressed only in the nuclei of a particular set of cells along the length of the worm. Okay. So, the worm has something called the seam, basically horizontal structure providing structural reinforcement for the body, and that is made up of 16 pairs of cells, and this particular gene is expressed only in those 16 cells. The rest of the 959 cells, they are all dark and only these glow green. Okay. So, but it was too late, so I did not make a picture that could be shown here today. So, but this is doing the same job. So, you see it is only in the lens, not even in the rest of the eye. So, because you do not want crystalline expressed elsewhere, you want only in the lens. So, now you are getting an idea of what is differential gene expression, right. Alright, now okay, so for studying the enhancers, now what we are doing is, can I use the enhancer to do something else, applications of enhancer. So, they are very useful to two different contexts we are going to see, two different examples. One of them is often used in mouse where you want to have conditional knockout. Let us say a gene has a crucial role in cleavage stage of the embryo, and if you just hit it, okay, null allele you create, then it is going to be embryonic lethal. Imagine that gene had a function in the liver and you want to know specifically what it does in the liver, you will never know that. So, therefore, when people want to find tissue specific or organ specific function of a gene, they want to have conditional knockouts, okay, meaning conditional deletion. So, this knockout at all slang that is being used so widely that it is also there in textbooks and we use them because we understand the meaning, but otherwise basically what you mean is deletion of a gene. That is what the non-geneticists initially called a knockout, and now geneticists also use it. Then some people call knock in, and then people say knock down, okay. A partial depletion they call knock down. I just do not like that word because that does not convey the meaning, it does not say what depletion tells. And similarly knock in, meaning transgene, and insertion they call knock in, okay. So, now we are looking at conditional knockout, meaning we are not deleting it at the zygotic nucleus. So, it is not going to be there in all cells, we are going to specifically knock out in a particular somatic tissue. So, how do you do that? How enhancers help? So, now you have one strain of mouse, okay, where you have a bacteriophage recombinase. So, okay, I tell you a little bit about the bacteriophage. I am not sure how many of you have learnt bacteriophage life cycle. Did anyone learn lambda-phage, lysogeny and lystic cycles? You know them. So, therefore, what do bacteriophage do for their living? What do they do? How do they survive? Host cell, like bacteria is the host. So, they get integrated, okay, and then when favorable conditions come, okay, from that itself they can make multiple copies. So, they can come out also. So, they can insert them and come out. How do? They do it using site specific recombination, and that is what is being exploited here. Because if I say recombinase and if you did not know what is recombinase, then there is no point in going forward. So, Cre is one such recombinase, okay. So, you express that protein under the control, in this particular example enhancer for albumin, okay. So, when you have a mouse strain that has Cre recombinase under albumin enhancer as a transgene, so now what this is going to do is it is not going to express anywhere except in the tissue, in this particular case liver, where albumin will be produced, only there Cre recombinase will be produced. Now, you have another mouse strain where your gene of interest, for example, in this case this transcription vector HNF4 alpha, its exon 2 is flanked by the sequence recognized by the Cre recombinase, okay. So, that is how the site specific recombination works, right. So, the site for the Cre is called, this is bacteriophage P1 and that is called LOXP sites. So, the LOXP site flanks exon 2. So, normally people call that as floxed, okay, flanked by LOX, so floxed. So, you flox the gene of interest. This exon 2 is not going to be removed in that strain anywhere, because there is no recombinase normally in the mouse that will recognize LOXP. So, that is going to be healthy, and simply expressing Cre recombinase in the liver is not going to cause any problem for that strain, because it will work only if there is a LOXP. Otherwise, you are producing a protein that is neither toxic nor having any functional interference. But if you cross these two mice and generate a double mutant, a mouse that carries both of them, in its liver you will have both the functional elements brought together. You will have Cre recombinase produced because albumin enhancer makes it expressed in the liver, nowhere else. And you need to remember, this is germline floxing. So, this is a line, it is a mouse strain. So, it is going to be there, this sequence is there in the genome of all the cells. We already know genome equivalence, and that does not matter, only in liver where you have the Cre recombinase, the LOXP site will be recognized and it will treat in between like bacteriophage genome and it will excise out, and then you will not have exon 2. So, this is conditional knockout. So, this is how people use enhancers, applications of enhancer, okay, one of them. So, now in a journal club or in a seminar, if someone says conditional knockout and I floxed this gene, you immediately understand what it is, right. So, it is very important to know this. And second one, this is primarily used in Drosophila, but it is extremely powerful and they came up with this for a long time ago, and only recently people have been successful doing it in C. elegans. Probably one, people did not find really important context where they needed it, another one lazy to develop a tool or whatever, but recently they have shown it works. But in Drosophila this has been very well exploited, and you know, this one example is good enough to know how powerful this is. So, this is GAL4 UAS system, very similar in you know, in principle to the previous one. You will have two different strains, okay. Here one strain under the enhancer of, you know, this a particular imaginal disc, okay. I do not know how many of you know imaginal disc. So, imaginal disc goes back to metamorphosis, okay. So, Drosophila being an insect, it has a worm stage, right, caterpillar sort of stage. From this the fly comes out, so there is metamorphosis. So in that worm like stage, it has the primordium for all the adult stages, and these primordia are what we call as imaginal discs. So you say wing primordia means these wing imaginal disc means the primordial cells that are present in the caterpillar, which will eventually be expanded into wing in the adult. So those are the imaginal discs. So in a very specific imaginal disc, or rather an imaginal disc specific enhancer, you use that to control the expression of an yeast transcription factor, GAL4. So GAL4 again has a very specific short sequence that it recognizes and activates transcription. So now you have that transcription factor produced in one strain of Drosophila under this imaginal disc specific enhancer. Now you have another strain where you have the in the upstream of your gene of interest, okay, or the gene itself is a transgene. For example, PAX6 you are taking from the mammalian system and putting it here. So you try to express this under the control of the promoter where GAL4 binds, okay. So this is called upstream activating sequence, therefore UAS. So the UAS here is GAL4 UAS, and that is for PAX6, and in the other one you have let us say I specific I imaginal disc, okay. So now when you cross both and make a double mutant, you are going to produce PAX6 protein where normally this will drive the GAL4 expression. So in this particular case it is not I, it is another structure in the thorax or in the head region, where by driving PAX6 they were able to induce the formation of I there, okay. A place probably antenna in this case, I think I have to check the book to see. Instead of developing antenna you develop I. So what it tells you here is PAX6 acts like a master regulator of I development. So once you provide PAX6, then looks like everything else is already there in that particular tissue to drive the formation of I. So we will see similar such complete organ formation in a wrong place, more examples later as we go through. A good group of genes are the Hox genes or the homeotic mutations, where you have one organ replaced by another organ simply because you changed the master regulator there. So it gives you versatility, like for example I can have this line and I can cross it with another line where the UAS is for another gene. It gives you that flexibility. So I can have a library, like driver is this and this is the UAS. So like that I can have, and then by bringing them together I can activate that. So that is the main reason. In the previous one also it is the same. So that is because you want to know in which imaginal disc, okay, if I activate PAX6 I will see this phenotype, or rather if I express PAX6 in different imaginal discs, what happens. So that is what you are testing. You did not get it. So see, you want to know like two things here. One is what does this protein do if it is put in a particular biological context. Another one is which imaginal disc has the ability to develop what kind of structures, okay. So normally it may develop, let us take this for argument antenna normally, but it has the ability to make I as well if you provide an I specific transcription factor. So that is one thing. Second, this also tells which imaginal disc can develop what structures. So both, it tells about the master regulator, like what this tells you is a necessary and sufficient condition for PAX6 to develop I, right. And the second is which imaginal disc is responsible for what structure. So both you can get out of this. So, this has been extensively exploited by the Drosophila developmental biologists. You will find lot of papers, you will hardly find a paper where they do not use this method. Alright, so that ends this, but we have not finished. So, we have another lecture we can begin, which is basically continuation of the same. Yeah, so we already saw this. Yes, so this kind of tests you, this will be the last concept for today. We are going back to the same PAC6 promoter region. Okay, so you need to remember this, I am talking about PAC6 promoter. So, where PAC6 expression, how it is regulated is the focus. So, we are looking at its own enhancer, and in turn PAC6 when it is produced, it itself is a transcription factor as well. So, that we will see in the next slide. So, I do not want you to get confused. We are looking at the promoter as well as protein of the same gene. So, now we are revisiting these different enhancers. Remember I told you each one seems to be a particular tissue specific expression. So, that is being tested here in this particular image, where you are only taking a part of it, like you are taking these two enhancers and the rest are not there, but you have LAGZ reporter here, and as a result you see in the pancreatic region as well as where the eye is developing, lens and cornea, only there you see, and you do not see in the if it is neural tube you should have seen in this part. So, you do not see it. So, this again demonstrates the modularity of the enhancers. So, in this particular gene it comes in 4 modules. Okay. When all 4 modules are there, it will be expressed in 4 different tissues, spatial regulation is conferred. Okay. So, this is modularity. Now, we are going to look at another concept of enhancer function, which is combination, okay, combinatorial function. So, that is there a little bit in this sequence, where what you are finding is to one particular module a combination of transcription factors bind, and only when these 2 transcription factors are binding to this pancreatic module will the PAC6 be expressed in pancreas. Okay. Transcription factors act combinatorially or in combination, and enhancers act modularly in a modular fashion, each module responsible for one specific tissue. So, that is further explained in this particular cartoon. So, we are looking at 2 different genes. Okay. In one, a target gene of PAC6. So, here we have gotten the PAC6 protein, and then we are seeing what it does to the promoter of another gene, it is a transcription factor. So, to make this particular crystalline expressed in the lens, PAC6 has to bind to its binding sequence, and that is not enough. It has to have a combination of SOX2 as well as LMAF. Only when all 3 are present, this crystalline will be expressed in the lens. This combination of transcription factors are required. Okay, that is why we say transcription factors work in combination. And these other proteins shown here aid in these interaction among these 3 transcription factors when bound to this promoter region. And additional example is here, somatostatin, the growth hormone. It is produced in pancreas, where PAC6 binds to its own binding sequence, but then that promoter should have other transcription factors, for example PDX and PBX binding to their own respective enhancer regions. Only when all 3 are there, this somatostatin transcription will happen. So, enhancers have modulus and transcription factors work in combination in each of those modules. Is this clear? So, I will stop here. Yeah, so this is the summary of what I just said. So, combinatorial functioning of transcription factors make coordinated gene expression. Okay. So, therefore, like for example, you might have under a particular condition you want to activate certain set of genes in certain parts, and all of them can be handled if that condition activates only one transcription factor. So, that will go to the right places, and if the right things are already there, it will express. So, that coordinated expression is possible. Okay. It is a very sophisticated coordinated expression compared to the yeast gal4 or bacterial operant systems, okay, where you have all the coding sequence under one promoter, but here you have different controls and more sophisticated coordinated expression. So, I will stop here. In the next class we will get into transcription factors. Okay.